Chromo Chromosomes is the thread-like structures. These are the self-replicating genetic structure of contain organized DNA molecules package found in the nucleus of the cell. Especially these are the found in the nucleus. Okay, these are the containing the DNA molecules. The next one is the self-replicating genetic material. What are the self-replicating genetic material? The DNA is the self-replicating DNA. Self-replicating material DNA. Next one, especially the seen the metaphase of the mitosis, especially the chromosomes are seen metaphase, mainly metaphase and prophase, metaphase, anaphase, telophase, especially these are the seen in the metaphase. Chromosomes seen during the metaphase stage, mitosis. The cells are the stained with the suitable basic dye and the view under the light microscope. Okay, especially these are in the section onion tips, especially in the onion tips, you can see the some of the in chromosome, especially in the mitosis space. Mitosis is clearly seen in mitosis metaphase, especially in the metaphase. Okay, in 1875, the discovered the Strasburger. 1875 discovered the Strasburger. The mainly these are the thread-like structures. Okay, mainly they are during the cell division in 1875. Strasburger discovered the chromosomes in the during the cell division. All type of the higher organisms. What are the all type of the organisms, especially in the eukaryotes? The prokaryotes is the lower, especially the nucleus, especially nuclear envelope is the absent. Okay. At the same time, some of the prokaryotes also they contain the 70S ribosome. Most of the prokaryotes is the lower type of the organism, and the eukaryotes are the higher type of the organism. Already eukaryotes they are containing the nuclear membrane, next one, some of the 80s ribosomes. So, okay, most of the cell organisms contain the prokaryotes. Okay, at the same time, these are the higher organisms, especially they are containing the well organized the nucleus contain different number of chromosomes. Okay, these are the special type of structure and the shape, size, the shape, size. The next one, uh, first one discovered by the Strasbourg in 1875. The Walder, the term is coined by the chromosome the first time in 1888. The chromosome, the term coined by the Walder in 1888. Okay. Next one is the somatic chromosome numbers, especially the chromosome, most of the diploid condition, the represented by the diploid 2n condition. Most of the somatic number of the chromosomes is the diploid condition is found in the somatic cells, especially vegetative cells is also called as the somatic cells. Mainly is the diploid condition. So somatic chromosome number is number of chromosomes found in the somatic cell and represented in 2n stage. At the same time, gametic chromosomes number also half of the somatic chromosome. What are the half of the chromosomes? Especially that indicate the 2n. Half of the mean X stage or N applied condition. Especially the diploid condition, they can see in the some of the somatic cells. In the applied condition also they can see in the only in germinate gametic cells. Gametic cells are the applied condition. Mainly two types of the chromosome and identically and morphologically gene content is known as the same two types of the chromosome. Ordinary the identical in morphological and gene content, gene order. There are also known as the homologous chromosome. Homo means same. Okay, the two copies of the chromosomes is ordinarily the same type, the homologous chromosomes. Hetero means different. One is the chromosomes are the types of the chromosome, two types of the chromosome. One is the autosomes. Next one is the sex chromosomes. Sex chromosomes. Okay, allosomes, autosomes, and allosomes. Allosomes is also called as sex chromosomes. Okay, autosomes are the control the characters of the other than the sex characters. Okay, approximately the human beings are there containing the 23 pairs of the chromosomes. The 22 is the autosomes. Okay, one single pair of the chromosomes is indicate the sex chromosomes, is allosomes. Okay, allosomes, autosomes, especially what are the other than the sex chromosomes. They are the controlling the other than the sex chromosomes. Carry the genes are the carrying for the, so especially they can carry to the somatic characters. Okay, only they can transform the somatic character. The sex chromosomes also involved in the sex determination. Mainly these are the XY condition, especially autosomes are the XX condition. Oh, 22 pairs. In 23 pairs, only one set of the chromosomes is the allosome. Sex chromosomes are gonosomes is also called as the gonosome. Chromosomes are involved in the sex determination. Humans and the most of the other mammals have two sex chromosomes. XY is also called as the heterosome. Already, this is the homologous condition. You can see in the some of the autosomes. Already, in the previous slide, we discussed the homozygous chromosomes, homologous chromosomes. The heterozygous is the heterologous chromosomes. Heterosomes are the XY condition. These are the, you can see in the human beings and the some of the mammals. Two sets of the chromosome X and Y also is also called as the heterosome. The females are especially they contain the XX. The XX mainly is of the females have two X chromosomes in the diploid cells. Males especially males they contain the XY chromosome. Okay. In the birds also, the female is the ZW condition, is the heterogametic condition. The male is the homogametic condition. Especially the females are the homozygous condition. In human beings, especially the females XX, the homozygous condition. Okay. Homogametic condition. Next one is the male are the XY condition. They are the opposite of the birds. The birds also they can see in the female condition is the ZW is the heterogametic condition. Male is the ZZ is the homologous condition. Okay. Okay. Next one is the chromosome number in the applied chromosome number. Already we discussed the applied condition, diploid condition. They can see in the most of the somatic cells. They are contained in the somatic cells. Okay. Diploid condition is indicate the 2N or 2X condition. 2N is the N is the chromosome number or X is the chromosome number. How to the homologous copy of the each chromosomes. Mainly are the body cells of the animals are the diploid condition. Bodies vegetative or somatic cells they are contained in the diploid condition. The applied condition, especially they are the seen the some of the gametes. Okay, some of the sperm cells are they are containing the sperms and the egg cells are the only applied condition. Okay, half of the condition in diploid, half of the condition the applied condition. The applied cells have the only one copy of each chromosome. In animals, the gametes, sperm and the egg cells are the applied condition. Chromosome number is the various from 2n equal to 4. Okay, n equal to 2. And next one, the aplopappus. Okay, in the aplopappus, they are containing the low number of the chromosome. And next one, ophioglasm 2n equal to 1260 chromosome. Okay, it's the ophioglasm is belongs to the pteridophyta. Ophioglasm is belongs to the pteridophyta. Okay, sorry for that. Okay. Diploid condition, especially diploid condition, they can see in the most of the gametic cells, the diploid condition, the somatic cells. Okay, sorry, somatic cells are contained in the diploid condition. And next one is the gametic cells are the applied condition. The aplopappus is the low chromosomes in the 2n equal to 2, sorry, 2n equal to 4. Actually, in the highest chromosome, they can see in the ophioglasm. Ophioglasm 2n is equal to 1260 chromosomes, they can see in ophioglasm. Ophioglasm is belongs to the Pteridophyta. Okay. Okay, next one. The number of chromosomes varies in the from species to species. That they can, for example, in rice, they're containing the 2n equal to 12 in rice, oryza. Okay. The women being 12 to 12 to 2n equal to 46. The different the species to species the various. Okay, the number of chromosomes varies from the species to species. Normally, all individual species have the same chromosome numbers. Okay, next one, size of the chromosomes, and next one is the shape of the chromosomes. Mainly shape of the size of the chromosomes is the measured approximately 0.25 micrometers in fungi. Okay, especially they can absorb in the mitotic division of the metaphase. Mitotic division of the metaphase is the approximately in fungi the short 0.25 micrometers in fungi. The boards is the long 30 micrometers in some of the plants like the birds, fungi and birds. Sorry, in the fungi birds also 0.25 micrometers. And next one, some of the plants are the size of the chromosome is the approximately 30 micrometers in like a trillium. Okay, each chromosomes are the two arms. One is the short arm and the long arm. Okay, two arm. Next one is the chromosome shape. Especially the chromosome shape, or they can absorb in the anaphase and in anaphase. Actually, the mitotic is in metaphase. They can absorb in the chromosome size. Okay, chromosome size they can mainly absorb in the mitotic the metaphase, and the next one is the chromosome shape. They can absorb in mitotic anaphase. Okay, anaphase mainly absorbed in the anaphase. The position of the primary construction centromere. The primary construction is also called as the centromere. It is the determine the chromosome shape, especially different types of the chromosome shape. V, L, J, I. We discussed the different types of the chromosome shape. Next one is the construction of centromere can be the terminal and the subterminal median in the position. Okay, okay. Chromosome shape also mainly the chromosomes are especially in the eukaryote cells are the DNA found in the component the nucleus especially. These are the together some of the proteins, and next one is the some of the molecules together they can form the chromatin. And next one is the what is the meaning of the chromatin? The DNA components the nucleus the form numerous linear molecules, plus next one some are the proteins together they can form to the make up the chromatin. Okay, chromatin. Actually, the short arm, the short and long arm also shape of the chromosome, size of the chromosome. The chromosomes, the two arms separated by the only the centromere. In between the centromere, they can contain the short arm and the long arm. The end of the chromosomes is the telomere. Okay, the end of the arms is also called as the telomere. The arms are divided into two. Each part is known as the chromatid. Chromatid, especially the arms are the divided into two, and each part is known as the chromatid. Already chromosome, different shapes of the chromosome. We discussed the L shape, next one V shape, I shape, metacentric especially is the centromere half of the long chromosome and two arms are the same length. These are the V shape. Okay, metacentric, the submetacentric. Submetacentric is the one arm is the longer than the other one. One division takes the L shape is the submetacentric. Metacentric is the V shape. Submetacentric is the L shape. Okay. Next one is the acrocentric. Acrocentric is the centromere is the very close to one end. The meaning of that is the arm is shorter one is then centromere. Okay, I is the centromere. Okay, next one is the centromere is the end of the chromosome. The chromosome only one arm. Okay, these are the shapes of the chromosome I shape. Okay, the telomere is the I shape. Okay, telomere is the I shape. Okay, the construction of the centromere can be terminal, subterminal, and the median position. Next one. Okay, homologous chromosome. Already we discussed the homologous chromosomes. Diploid organisms have the two copies in each chromosome except the X chromosome. What are the except the X chromosome? XY chromosome except the XX and the XX chromosome. Diploid organisms have a two copies of the each chromosome. The both the copies are ordinally, identically, morphologically, and next one is the content the same structure, identically, morphologically same. The same structure is also called as the homologous chromosomes. Each pair of the chromosome made up to two homologous chromosomes inherited from the separate parents. Homologous from the mother and the other comes from the father. Okay, homologous chromosome. One is from the father, one is from the mother. These are the species, different types of the species, and the number of chromosomes in human beings, especially 2n equal to 46. Okay, and next one is the cat 38. Okay, dog 78. Next one is the horse 64. Next one is the uh, ostrich also there contain the 2n equal to 80 chromosomes. Ostrich. Next one is the same Oryza sativa 2n equal to 12. Oryza sativa. Okay, next one is the most of the Graminea 2n equal to 4. These are the number of chromosomes, size of the chromosome, number of them. Next one, honeybee actually is the honeybee is female opinion. The workers have the 32 chromosomes, and the male is also called. They contain the drone. You have the 60 chromosomes. 60 chromosomes. Next one, chromosome morphology. This one is very important. The especially chromosome morphology, mitotic metaphase is the most suitable stage of the studies of the chromosome. Especially, already we discussed the size and shape. Size, especially, they can study in the in mitotic in the metaphase. Okay, and next one is the shape. They can study in the mitotic anaphase. Size and shape. Sorry, shape and size. Shape, especially, they can study in mitotic region in the metaphase. The next one is the size, especially, they can study in the anaphase. Chromosome morphology. Metaphase is the most of the suitable stage studies of the chromosome morphology. The chromosome morphology changes during the cell divisions. Okay, chromosomes are the what are the chromosomes are the thin, are the coil, elastic condition, the thread-like structure. These are the during the interphase. During the interphase, these are the mainly chromosome morphology. The changes during the cell division. Chromosomes are the very thin and the coil nature, elastic, the thread-like structures during the interphase. Thread-like structure during the interphase. Next one, the cell entering the mitosis, the chromosomes become highly condensed that the can be distributed the daughter cell and the mitotic metaphase chromosome. These are the following some are the features of the chromosome morphology. So these are the morphology. The satellite, these are the satellite chromosomes. Okay, end of the telomere. These are the arms. Both the arms, the short arm, the long arm. Okay, both are the connected in the centromere. Is also called as the primary construction. The centromere is also called as the primary construction. And next one is the end of the portion is also called as the telomere. Okay, these are the telomere. Next one is the in between the some of the secondary construction also they can be seen in the secondary construction. The primary construction and the secondary construction they can see in the chromosome morphology. Next one, chromatid. What is the meaning of the chromatid? Chromatid mean especially each metaphase chromosome appears to the longitudinally divided into two identical parts is also called as the chromatids. These are the chromatids. For example, these are the chromatids. The both are the chromatids. Okay, these are the in the each identical part. They can divide the equal position. Okay, the metaphoric chromosome appears the longitudinally divided into the identical parts, which is also called as the chromatids. These are the chromatids. See, this one is the these all are the these are the chromatids. The both are the chromatids. Okay, the both the chromatids chromosomes appear to the join together the centromere. They are the centromere. The centromere attached to the both the chromatids. Okay, these are the chromatids. These are the centromere. Centromere also called as the primary construction. Chromosome. Next one is the two chromatids of the chromosomes separated from the each other. Mainly the centromere. Centromere. Okay, next one is the two chromatids make up the chromosomes are the produced through the replication of the single chromat during the synthesis S phase of the interphase. The formed the sister chromatids. The both are the same sister chromatids. Mainly the synthesis of the chromosome. Synthesis of the chromosome in S phase of interphase. Next one, some of the contrast chromatids of the homologous chromosomes are known as the non-sister chromatids. Sister chromatids is the heterozygous, the heterozygous condition. Sister chromatids, non-sister chromatids is the homologous chromosomes are the also chromatids of the homologous chromosomes also known as the non-sister chromatids. Next one, centromere. Already we discussed the primary construction of the centromere. Centromere is the mainly landmark identification of the chromosomes. The chromosome construction point is also called centromere. Its synonym is the kinetochore. Is also called as the kinetochore, which is the divides the chromosomes, the two sections of two arms. The two arms are also called the chromatids. The both the chromatids are the attached from the centromere. The both the one is the short arm and the long arm chromatids also they can see in the in chromosomes. In the short arm of chromosomes labeled the P arm and the long arm is also called as the P arm. These are the different. Next one, telomere. Telomere is the two ends of the chromosomes are known as the telomere. End of the chromosome, for example, these are the end of the chromosomes. End of the chromosomes also called as the telomere. End of the chromosomes also called as telomere. End of the chromosomes also two ends of the chromosome known as the telomere. Mainly these are the they can critical roles of the chromosome replication and maintenance of the chromosomes length. The telomeres are the highly stable the telomers of the different chromosomes. So telometric region chromosomes made of the repetitive sequence of the T and G bases, thymine and the guanine bases. Next one, secondary construction. Already the primary construction is the centromere. The secondary construction, some of the chromosomes addition to centromere, is also called as the secondary construction. Mainly these are the more construction in the chromosomes are the present term is the secondary construction. The number of construction, the primary, more than one connection, the primary construction is also called as the secondary construction. A satellite end of the position, the chromosomal region between the secondary construction and the nearest telomere is also called as the satellite. These are the satellites. These are the satellite nearest the secondary construction, nearest the telomere is also called as the satellite. The chromosomal region between the secondary construction and nearest to telomere is also called as the satellite chromosome. These are the process and the region called is the satellite chromosome or SAT chromosome. Satellite chromosomes and satellite chromosome. A small segment separated by the main body chromosome, the secondary construction is also called as the satellite. Mainly the secondary construction. The secondary construction separated by the some of the chromosomes. Mainly the small segment separated from the main body, the chromosome. Secondary construction is also called as satellite. Next one, the of the chromosomes. Size of the chromosomes is the various stages cell division. The mainly are the chromosomes are the longest thickness during the interphase. The interphase is also called the resting stage. These are the not visible under the light the microscope. Mainly these are the size of the chromosomes. The chromosomes are the longest and thinnest during interphase. They are all resting phase. They cannot see into under the light microscope. Especially they are seen to the electron microscope and the phase contrast microscope. Different types of the different types of the microscope they can see in the chromosome size. Chromosome size is not proportional to the number of genes present in the chromosome. Mainly the location of the centromere each chromosome gives to the chromosome the characteristic shape especially. Okay, the location of the centromere on each chromosome gives to the chromosome the characteristic shape. These are the chromosomes classified according to the centromere position. At the end of the acrocentric, okay, chromosomes are classified according to centromere position. At one end of the acro mean end, acro end. For example, this is the acro end. Next one is the closer to end, submetacentric, submetacentric. The submetacentric, especially J shape. Submetacentric is the J shape. The middle V shape. Metacentric is the V shape. Each chromosomes have the two arms. One is the longer arm and the shorter arm. Longer arm is the indicate the Q and the shorter arm is to indicate the P. Okay. P arm is the petite and the small is the meaning of the small P. P means small. Q arm is the mainly the follow the P is the long. P is the long. Sorry, P is the short and the P is long. Metacentric chromosomes. What is the meaning of the metacentric chromosomes? The centromere is located in the center of the chromosomes. Meta means center. Meta means center. Especially centromere is located. For example, these are the V shape. Okay, these are the metacentric. Metacentric. Especially the centromere is located in the center of the chromosome. That is the chromosomes is the median. The centromere is the localized the approximately midway between each the two arms is roughly equal to the length, same length. See, these are the same length. The chromosome chromatids are the same length. When these are the V shape, metacentric. Metacentric are the indicate the V shape during the anaphase. During the anaphase. Next one, suburb metacentric. The centromere located on the one side of the central part of a chromosome. These are the J shape, J shape or L shape, J shape or L shape. One arm is the longer and another arm is the short arm. The submetacentric condition is the indicating the JL shape during the anaphase. During the anaphase. Acrocentric, already we discussed the acrocentric end. The centromere located at the close one end of the chromosome. Acro, telo end. Acro is the mainly the acro may refer to the peak end position. The centromere is more terminally placed in the forms of the very unequal form from the length. Acro mean acro refer to the Greek word the peak. Acrocentric chromosome made rod shape during the anaphysis. The acrocentric, these are the series on acrocentric. Okay, acrocentric, especially telocentric and the acrocentric, the eye shape. They can indicate the eye shape. Okay, eye shape. Telocentric. Telocentric is the centromere located in the end of the chromosome, the terminal position. Okay, this is also you can see in the anaphase. Okay. Mainly these are the according to number of centromere in eukaryotic chromosomes made divided to the eccentric. Eccentric mean without any centromere. Eccentric. Okay. Next monocentric mean only one centromere. They're contained in the only one centromere. Next one is the dicentric. Dicentric. Sorry, dicentric mean two centromeres. There are the two centromeres. The polycentric with a more than two centromere. Eccentric mean there is no centromere. Monocentric mean within the one centromere. Dicentric mean with the two centromeres. Next one, polycentric mean with the more than two centromeres. More than two centromeres. Okay. The according to number of centromeres, the four types: the eccentric, monocentric, dicentric. Poly mean many centric. Okay. Next one is the sequential DNA. Okay. DNA plus histone proteins is also called as the chromatin. Especially histone proteins are the they can see in the most of the in eukaryote cells. The histone proteins are the absent in prokaryote cells. Okay. The DNA double helix is the cell nucleus packed by the special proteins termed as the histones. Okay, the form the protein DNA complex is also called as the chromatin. Chromatin means DNA plus histone proteins is also called as chromatin. DNA plus the histone protein is also called as chromatin. The structural entity of the chromatin is the nucleosome. The chromatin entity is the nucleosome. Nucleo mean especially nucleus, nucleosome. The chromatin, nucleosome, and the DNA bonding, and the finally form the DNA. Chromatin. Chromatins are the what are the chromatin? Histone plus DNA is also called as chromatin. Histone and DNA is also called as chromatin. Chromatin is the indicate the nucleosomes. Nucleomine especially is the some of the genetic materials they contain the nucleus. Nucleus are occurs in the some are genetic materials. The DNA binding proteins. DNA. What are the DNA binding proteins? Histones. Then next one DNA. The DNA plus DNA binding proteins are the histone. This is also called as chromatin. The chromosome packing. The chromosome packing. Let we discuss the stones the group the five major classes H1 to H5, H1 A to H2B H3 H4 the five type of the protein. They are the organized into the two super classes following to the H1 and H1 and the H5 is the main linker histone protein. The core histones is the H2 H2B H3 and H4 the core histone proteins. They are the histone proteins are in the some of the chromosome. Okay, nucleosome. Already we discussed the nucleosome. These are the basic unit of the DNA package in the eukaryotes. These are the basic unit of the DNA package in eukaryotes. Basic units of DNA package in eukaryotes. The consist of the segment of DNA, especially basic unit of DNA package is the eukaryotic nucleus. The consist of the segment of DNA around the histone protein. The functional repeating units the eukaryotic chromatin. Actually, the nucleosomes are the core particle consists of the approximately 147 base pairs of the DNA wrap. Actually, we just the five types of the histones. Okay, the nuclear cores are the coil into the solenoid shape itself. The coils of the further complete of the DNA. These are the solenoid steps. These are the solenoid steps. Especially, these are the how many histone proteins are the same. These are the five histone proteins are the same. These are the solenoid steps. Okay, mainly linker DNA. Some are the code particles connected to the structure of the linker DNA, which is the approximately can be up to the 80 base pairs long. Okay, linker histones also there H1 and the it's involved the chromatin compensation of the nucleosomes near DNA and the existing binding of the region of the DNA. Non-condenser nucleosomes, condenser nucleosomes and non-condenser nucleosomes mean without the linker histone resemble the beds of the string DNA. Okay, they are under seen the electron microscope. Already we discussed the chromosomes also only they can see in the electron microscope. Already in the previous slide they cannot see in the light microscope. The DNA, the most of the chromosomes they can see in the the electron microscopes. Non-condenser nucleosomes mean without the linker histone resembles. Okay, without the condenser nucleosomes mean with the linker histone resembles the condenser nucleosomes. Next one is the protein DNA structure chromatin stabilized with the attachment of the non-histone proteins, the scaffold called the nuclear matrix. Nuclear matrix. Most of the eukaryote cells the mature sperms of the large is the proteomics and the package of the reasoning. Histone equivalent to the simple and the chromatin structure have been found the archaea. Archaea means the ancient. Some are the histone, especially equivalent to the simplified chromatin structure have the also been the found in the archaea, providing eukaryotes not in the organism that is the nucleosome. Next one is the heterochromatin and euchromatin. What is the mean of the hetero mean different? Okay, hetero means the different. These are the tightly packed chromosomes. And next one is the consist genetically inactive satellite sequence. The both the centromeres and telomeres are heterochromatic. Both the centromeres and telomeres are the heterochromatin. Next one is the euchromatin. Euchromatin is the lightly tight, lightly packed. These are the heterochromatin mean tightly packed of the chromosomes. Heterochromatin is the tightly packed of the chromosomes. Okay. Next one, euchromatin means the lightly packed of the chromosomes. Lightly packed of the chromatin are DNA, RNA, and the proteins. Chromatin means already we discussed the DNA plus histone proteins. It's also called as the chromatin. Mainly in euchromatin is the tightly packed chromatin that is the rich of gene concentration. Okay. Mainly unlike the heterochromatin is found in the both the cells of the eukaryotes and the some of the especially nuclei of the eukaryotes and the nuclei of the prokaryotes. Prokaryotes is the especially without the nuclei. Okay, there are the heterochromatin and euchromatin. Heterochromatin and heterochromatin are the two types of the constitutive heterochromatin and the facultative heterochromatin. Constitutive heterochromatin is the presently two homologous chromosomes is the inherited within the chromosome, especially within the chromosome found specific regions. The term of they are the centromeric near to the centromere. The second one is the telomeric. Centromeric means near the centromere. Heterochromatin. Heterochromatin both is the mainly the interface, especially they can see in the interface nucleus in the contain dispersed chromosomes in matter contain the cold some of the especially age in 1925 28 kind by the heterochromatin denote the chromosomes region especially. remain the highly condensed state and the less stained regions. Highly condensed is the heterochromatin and the less condensed is the euchromatin. Okay, the regions showing the heterochromatin, the two types of the group, one the constructive heterochromatin, next one is the facultative heterochromatin. What are the functions of the heterochromatin? Mainly these are the heterochromatin leads to the different functions of the organs. That the main function is the uh, these are the temporary genetic activities, such as the unnecessary genes are maybe larger the functional abilities. Okay, functional like the especially they are the seen to the evolutionary periods of the some of the organisms like insects. Some of the insects. Next one, heterochromatin is the genetically addition of the or loss of the heterochromatic segments does not appreciably affect of the phenotype studies in the Drosophila. Heterochromatin, the heterochromatin, solenoid model. Next one, models of the DNA for folding the two, according to our syllabus, two models is there: nucleosome model and the solenoid model. Mainly the models of the DNA folding discovered into the electron microscope, 1960s. Mainly is the 1960s. One is the nucleosome model. Mainly nucleosome model is the electron microscope studies the chromatin from the interface nuclei. Uh, the spherical particles are the approximately 10 to 1 nanometer equal to 10 to the power of minus 10 angstrom, minus 9, sorry, 9 minus 9 angstrom. Repeating of the sum of units which is occur regular intervals given the specific is also called as the nucleosome. Mainly nucleosome models are they contain the five histone proteins H1, H2A, H2B, H3, and H4. How many proteins are the histone proteins? Five. Especially in nucleosome model, they are containing the five histone proteins. The five histone proteins. Mainly the cross-linking of the histone proteins are the found in H3 and H4. These are the present in the tetromere. Tetromere. The next one is the some of the length of the DNA is the approximately 2,200 base pairs. 200 base pairs. The DNA is the present between the adjacent core particles vary from 18.8 to 111 114 base pairs and is the link to the DNA. Mainly, okay, the nucleosome model, nucleosome especially they contain the how many histone proteins? Five types of the histone protein. The solenoid is the approximately the 11 nanometer fiber nucleosomes gets coiled up and form 30 nanometer wide helix. 5 to 6 nucleosomes for the helix. Each helix are there containing the solenoid model. The solenoid model is the approximately 11 nanometer fiber of nucleosomes gets coiled to the itself from 30 nanometer. What helix the each helix they're containing the 5 to 6 nucleosomes. Okay, so each helix are there containing the 5 to 6 nucleosomes. The 30 nanometer structure was the also called as solenoid. The top solenoid is the physics term. Is the called the central axis. Central axis. They are the solenoid model. Okay. Euchromatin and heterochromatin. Next one is the special type of the chromosome. Okay, next one is the special type of the chromosome. But this was the chromosome structures. Okay, special types of the chromosome. Some of the organisms are they contain the special tissues in the which chromosomes undergo a special structure. What are the special type? Especially mainly the special type of the structures are they can see into the in Drosophila, oocytes of the amphibians. Some are the vertebrates also. Special categories called beta chromosomes reported in the some of the plant species also. Different types of the special types of the chromosome. Such as special type of the chromosomes including the giant chromosomes, polytene chromosomes, Lamb-breast chromosomes, or supernumerary chromosomes. The special type of the chromosomes. Special type of the group. What is the meaning of the giant chromosomes? Giant chromosomes is the exceptionally enlarged in the highly specialized chromosomes, the giant chromosomes. That are found in the some stage of the life cycle of certain cells. Mainly two kinds of the giant chromosomes. These are the exceptionally enlarged and the highly specialized chromosomes, giant chromosomes. These are the two types of the giant chromosomes. Endomitosis, okay, 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 two types of the chromosome. Mainly, these are the size of the results of the peculiar kind of the chromosomes reduplication called as the endomitosis and endoduplication. Endoduplication, the duplication of the and some of the cells, some of the size results of the peculiar kind of the chromosomal reduplication is also called as the endomitosis, endoduplication. Mainly DNA of chromatids, DNA of chromatids keep on the multiplying the geometrical provisions. The endomitosis makes especially chromosome multi-stranded and the increase its DNA content nearly 1,000 times of the more, 1,000 times of the more giant chromosomes. Next one is the giant chromosomes is the one type of the special and the polytene chromosomes. Polytene chromosomes also is the special type of the chromosomes. Mainly special type they can see in the interface of the mainly Sorry. Okay, polytin chromosomes is one type of the special type of the chromosomes. Is also they can see in the interface. The giant chromosomes are the found in the some of the cells of the salivary glands, especially in Drosophila. And Drosophila, mainly these are the giant chromosomes. The one polytin chromosomes and giant chromosomes they can see in the Drosophila. The This is the first observed in the salivary glands of the Cyronomus, and this is also called the salivary gland chromosomes, also called as the salivary gland chromosomes. These are the capable like the multi-stranded giant chromosomes. Under comes the polytin chromosomes also there come to the giant chromosomes. Okay, giant chromosomes. Okay, these are the multi-stranded giant chromosomes. These are the mainly, especially these are the found and discovered into the salivary glands of the some Drosophila, and next one is the Circonus larvae. Okay, mainly these are the polytin polytin mean many. They are also seen the synergy and antipodal cells of the ovules, certain angiosperms. Okay, in polytin chromosomes also they can see in the synergies antipodals of the some of the ovules, certain angiosperms. The first observed by the Balbani. The polytin chromosomes are the first observed by the Balbani. Balbani in 1881. In 81, these are the mainly some of the salivary glands of the insect or the diphtheria, the mosquitoes, and next one is the fruit fly Drosophila. The first one, okay, the polytin in 1881, Balbani. Structure of the polytin chromosomes, these are mainly the distinct pattern of the transverse banding. It consists of the dark color bands alternating with the light color interbands. Consists of the dark color bands alternating with the light colored in the interbands. Okay, nearly 85% of the chromosomal DNA is the dark bands. In the interbands is the 15%. Approximately, mainly the structure of the polytin chromosomes are they are contained in the two types of the bonds, dark bonds, and next one is the colorless, the interbands, and next one is the dark bond. Interbands is approximately the light color is approximately 15%. Okay, in chromosome DNA they contain the 85% of the dark bonds, so they contain the 80 dark band, and the 15% is the interbands. The mostly is the heterochromatic interbands, and especially you can see in the euchromatic condition interbands. Okay, the interbands is euchromatic mean lightly. Okay, interband is the lightly, and next one is the dark band. This is the heterochromatic condition. Euchromatic condition they can see in the interbands. In the heterochromatic condition is they can see in the tightly bands. Heterochromatic condition. In meiosis, in homologous chromosomes also they can see in the band to band condition. Each band is consist of the genes that can be is the more than one protein, and the polygenic condition. Each band they are containing the more than one proteins is the condition also called as the polygenic condition. Polygenic condition. These are the giant chromosomes. See the X chromosome, Y chromosomes are the giant chromosome diagram. Polytin is the mainly polytin chromosome. Okay, these are the mainly you can see in the chromonas, tetanus, salivary. These are the you can see in the larval salivary glands of the chromonas, tetanus, and the Drosophila. Okay, and these are the development of the some of the chromosomes, especially polytin chromosomes. Each bond believed that contains a specific type of the genome. They are containing the specific type of the genomes. The next one, each gene in the band have the multiple copies are the transcriptionally active in the same time. Next one is the structure of giant chromosomes are the modified to certain regions such as the one or more specific bonds is the enlarged and the plug out. And next one is the some of the forming local swellings is also called as the chromosome puffs. Chromosomes puffs mainly are the swellings, especially the forming local swellings, and is also called as the chromosome pumps. The formed uncoiling chromatin filaments. The uncoiling chromatin filaments. Some of the cases are the addition to RNA and
ఇది అలాగా మీరు అన్మ్యూట్ చేసి అలాగొస్తుంది. ఇప్పుడు హాఫ్ అన్ అవర్ అయిపోయింది. అతను చేస్తా
ସେ ଜାଣିନି। Someone video is on, Lalita Kumari.
Sorry for the disturbance. Actually, my laptop is switch off. Already we discussed the polytene chromosomes, giant chromosome. Next one is the Lamprecht chromosome. These are the mainly the found. The first one is observation of the Fleming in the 1882. Establer is the biggest algae. It belongs to the chlorophyce members. Okay, these are the Lamprecht chromosomes also. These are the special type of the chromosomes. Okay, mainly these are the observed in the some are the oocytes of amphibians. Next one is the spermatophyte nuclei of the insects, including the Drosophila, Stableria. Mainly these are the first one observed by the Fleming in 1882. Shark oocytes in the next one. Another scientist name is the Roquette. Also, they can observe in the some of the oocytes. Next one is these are the chromosomes showing the chromosomes pattern with the pairs of loops of projecting out from the majority of the chromosomes. These are the two forms of the bivalents with the maternal bivalent and the paternal homologs being is the chiasmata. It represents the low granules of the axis. Each homologous from the which lateral loops is extend the mainly these are the these are the lamb-brush chromosomes. See, these are the lamb-brush chromosomes. Okay. These are the loops always symmetrically with the pair of loops in the each chromosomes. And next one, these are the additional symmetrical pairs of the together they form to the homologue. Mainly these are the characteristics in the appearance of the chromosome is the seized by the rocket name is the lamb-brush. That the name is given by the lamb-brush chromosome. Mainly these are the name is given the rocket in lamb-brush chromosome. These are the look like the brush. The look like the brush is using those days the clean up the cinema of the oil lamps. Mainly is the look like the brush like structure is also called as the lamp brush chromosome. The name is given by the rocket. The scientist name is the rocket. These are the size of the loops are the various the average approximately 10 micrometers with the approximately 9 to 1 to 9 such loops are the each chromatic chromomere each chromomere. The loops are the constant position in the chromosomes. There in the many approximately the 10,000 loops for each set of the chromosomes. Okay, 10,000 loops for each set of the chromosomes. The loops are the site active genes and contain the RNA. Each loop is the form to the single DNA molecules the unfold from the chromosomes during the RNA synthesis. Mainly is the RNA synthesis is the very important RNA synthesis. Each loop covered is the matrix contains the RNA and next one is the transcripts with the proteins attached to them. RNA and the transcripts attached. Transcripts is the proteins attached to the loop. Next one, mainly RNA synthesis start from the thinner end of and progress toward the thicker. The structure is also called as the Christmas tree. The like structure is also called as the Christmas tree is a pattern. Next one is the many loops are the correspond the single transcription end of the continuous tick in the matrix. The loops contain the several units of the transcription. Mainly is they contain the RNA. RNA is related to the transcription. Okay. So some are the salivary glands, the chromosomes, the lamprey chromosomes are the mainly you can see the material for the studies of the in situ DNA hybridization. Salivary glands are the chromosomes and the lamprey chromosomes are they can use for the material for the studies of the in situ DNA hybridization. DNA hybridization. And next one is these are the lamprey chromosomes and next one is the Haltin chromosomes and next one another one is the beta chromosomes is are accessory chromosomes. Beta chromosomes is also called as the supernumerary chromosomes. Is also called as the supernumerary chromosomes. These are the mainly is that observed in maize, Longley and the Rodolfo in 1927. In mainly is are the beta chromosomes, especially the addition of the 2n equal to 20 chromosomes is the complement. These are the beta chromosomes. These are the observed in the during the meiosis metasis divisions. There are different types A chromosomes. Next one is the most of the beta chromosomes are they can see in the pollen fertility. These are the significant effect of the beta chromosomes on the pollen fertility and the seed set. At the same time, the flowering generally is the delay of the ray plant flowering delay. What are the important? What are the significance of the beta chromosomes? Is the mainly is the related to the pollen fertility and the seed set, the delay of the flowering. And next one is the number of beta transaction has been reported. Already beta chromosomes also studied the some maize, Rodolfo, and the next one Longley. These are the many important is another one. Another important is the mainly role of the plant breeding. Okay, plant breeding also they can plant breeding also they can key role of the beta chromosomes. These are the beta chromosomes. Is also called as the accessory chromosomes or the supernumerary chromosomes. What are the special type of the chromosomes? We discussed the mainly Pauline chromosomes, Lambrecht chromosomes. Next one is the beta chromosomes. Okay. Next one is some are the salivary glands chromosomes. Different. These are the different types of the special type of the chromosomes. We discussed the this class types of the chromosomes, and next one is the special type of the chromosome. Two lessons is over in this chapter. According to in the annual examination point of view, you will ask to the Pauline chromosomes, special type of the Pauline or Lambrecht chromosomes. These are the very important. Write an essay of the special type of the chromosomes. What are the special Pauline chromosome and Lambrecht chromosome? These are the very important question in the annual examination point of view. Okay, thank you.